A vaccine is a biological preparation designed to confer immunity or protection against specific infectious diseases. It works by triggering the immune system to produce antibodies and create memory cells. These memory cells remember the pathogen, allowing the immune system to respond quickly and effectively if the person is exposed to the pathogen again. The main goal of vaccination is to build immunity and prevent disease without actually causing the illness. There are several types of vaccines, each tailored to stimulate the immune system in a unique way. Let's explore these different types and understand how each one works. Live attenuated vaccines contain a weakened or attenuated version of the actual virus or bacteria responsible for causing the disease. The pathogen is altered in a lab to reduce its ability to cause illness, but it remains strong enough to stimulate an immune response. Because the immune system treats this weakened pathogen as if it were the real infection, it mounts a defense by producing antibodies. This process closely mimics how the body would react during a natural infection, leading to strong and often long-lasting immunity. These vaccines generally provide immunity with just 1 or 2 doses, as the immune response is robust. However, since the pathogen is still alive, though weakened, live attenuated vaccines are not recommended for people with weakened immune systems, such as those undergoing chemotherapy, living with HIV, or taking immunosuppressive medications. Even a weakened form of the pathogen could cause serious illness in these individuals. Examples of live attenuated vaccines include the measles, mumps, and rubella vaccine, and the varicella or chickenpox vaccine. Inactivated vaccines are made from viruses or bacteria that have been killed or inactivated, usually by heat, chemicals, or radiation. This process ensures that the pathogen cannot replicate or cause disease, making these vaccines very safe, even for individuals with weakened immune systems. Because the pathogen is inactive, the immune response is not as strong as with live vaccines. As a result, inactivated vaccines typically require multiple doses or booster shots over time to build and maintain adequate immunity. While these vaccines do not offer lifelong immunity like some live vaccines, they are essential for protecting against diseases where live vaccines might be unsafe or ineffective. Examples of inactivated vaccines include the inactivated poliovirus vaccine, the hepatitis A vaccine, and the rabies vaccine. Subunit, recombinant polysaccharide, and conjugate vaccines. These vaccines use specific parts of a pathogen, such as its proteins, sugars, or outer shell, to stimulate an immune response. Rather than using the entire virus or bacteria, these vaccines isolate and present only the essential parts that the immune system needs to recognize the pathogen. Since they do not contain live pathogens, these vaccines carry no risk of causing the disease, making them especially safe for people with weakened immune systems, such as those undergoing chemotherapy or with conditions like HIV. However, because these vaccines target only certain parts of the pathogen, they may sometimes require additional doses or boosters to ensure long-lasting immunity. Examples include the hepatitis B vaccine, the pneumococcal conjugate vaccine, the human papillomavirus vaccine, the Haemophilus influenza type B vaccine, and the meningococcal conjugate vaccine. Toxoid vaccines are specifically designed to protect against diseases caused by toxins released by certain bacteria rather than targeting the bacteria themselves. Some bacterial infections, like tetanus and diphtheria, are dangerous not because of the bacteria directly, but because of the harmful toxins they produce. To make a toxoid vaccine, the toxins are inactivated using chemicals or heat, rendering them harmless. These inactivated toxins, known as toxoids, are then used in the vaccine to stimulate the immune system. Although the toxoid cannot cause disease, it teaches the immune system to recognize and respond to the toxin if exposed in the future. When the body encounters the actual bacterial toxin, it can quickly neutralize it, preventing the associated illness. Examples of toxoid vaccines include the tetanus vaccine and the diphtheria vaccine. mRNA vaccines represent a modern approach to vaccination. Instead of using a live or inactivated virus, these vaccines utilize synthetic mRNA, a type of genetic material created in the lab. The mRNA contains instructions for cells to produce a specific protein from the virus, such as the spike protein seen in COVID-19. Once the vaccine is given, the synthetic mRNA is taken up by the body's cells. Inside these cells, the mRNA serves as a blueprint, directing the cellular machinery to produce the viral protein. The protein is displayed on the cell surface, prompting the immune system to identify it as foreign. The immune system then responds by creating antibodies and other defenses to target the protein. Examples of mRNA vaccines include the Pfizer-BioNTech COVID-19 vaccine and the Moderna COVID-19 vaccine. Viral vector vaccines use a harmless modified virus, known as the vector, to deliver synthetic genetic material into cells. The vector virus is modified so that it cannot cause disease or replicate within the body. Its role is solely transportation. Once inside the cells, the genetic material carried by the vector provides the instructions to produce a specific protein associated with the pathogen. This protein is then recognized by the immune system as foreign, prompting an immune response. Examples of viral vector vaccines include the Johnson & Johnson COVID-19 vaccine and the Oxford AstraZeneca COVID-19 vaccine.